Hello, everyone, and welcome to today's live broadcast, Strategies to Achieve Robustness in Clinical Proteomics Research, presented by Dr. Michelle Hill. I am Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Agilent. For more information on our sponsor, click on the sponsor logo on the left side of your screen or visit agilent.com. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Also, please notice, you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's live event. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located at the far left of your screen. I would now like to introduce today's presenter, Dr. Michelle Hill, Associate Professor, QIMR, Berghofer Medical Research Institute, Brisbane, Australia. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Welcome, Dr. Hill. You may now begin your presentation.
Thank you very much, Christy, and thanks everyone for joining me today. I'd like to share my experiences on the strategies to achieve robustness in clinical proteomics research. So firstly, before we dig into the details, let's just have a brief consideration on why we want to do clinical proteomics research. So we all know that genes makes proteins, and there are many different techniques now that enable us to measure the genome and the transcriptome, and they are well used. So is there really a need to directly measure proteins? Well, actually, yes. So proteins are the direct disease targets for diagnostic tests as well as for therapies. And there are many tests now that actually use proteins. So between the gene level and the protein level, there are many different steps, and there are many studies now that report they don't directly translate. So indeed, there is a real need to directly study proteins. And furthermore, there are many modifications that occur at the protein level that cannot be predicted from the genome or the transcriptome. So there is an actual need. And let me share an actual example with you so it's a bit more clear. So the example I wanted to use is the use of HER2 inhibiting drugs in HER2 positive or HER2 amplified cancers. Now, there are many different tests that one can use to measure the amplification of HER2. So for example, you can use in situ hybridization to detect the gene amplification. And as shown on the slide here, there's a method, and it detects this amplification in the nucleus. On the other hand, you can also use immunohistochemistry to detect the protein. And because it's a receptor, it's on the cell surface. And now sometimes it's important to detect the actual protein that's on the cell surface, because in certain cases, the protein may not actually make it to the cell surface. It may be mislocalized inside the cell, in which case the HER2 targeted drugs will not really work because the target are not on the cell surface. And a third method to detect HER2 amplification is actually a completely proteomic method. Because HER2 can be shed into the serum, and that's a quick, minimally invasive way to detect excess or amplified HER2 by measuring the protein in the serum. And so this method is actually completely inaccessible to genomic techniques. So I hope with this example, you can see that there is a need to directly study the proteins, and it can become useful in clinical practice. So with that introduction, let me first then define the terms that we're using so we can be all on the same page. So proteomics is the large-scale study of the proteins, and the proteome defines the whole set of proteins produced in an organism, a system, or biological context, and that's thought to be the protein complement of the genome. But of course, that the proteome includes differential spliced proteins, truncated proteins, post-translationally modified proteins, so it's actually a lot bigger than the genome. And to study this big complex proteome is rather challenging, and hence our webinar today to discuss how we can make the measurement of proteomes much more robust for clinical research. So the learning objectives today I'd like to go through are to consider the types of quantitative proteomics techniques, consider the challenges and the specific points which can lead to irreproducibility or variable data, and then the possible strategies that we can use to improve it, including using an example from our own research. So now let's consider the type of clinical research studies that we might use with proteomics. So quite often this is to find biomarkers. So I'll use clinical research study designs with biomarkers as an example. And on this slide, I'm showing three possible clinical research study designs. So the one on the most left is possibly the simplest. So one does a clinical research trial with a drug and a placebo, and afterwards one does a clinical proteomics analysis of the collected samples and come to the analysis where we can say patients with particular subjects with particular set of biomarkers are responsive to this drug, whereas a different profile of biomarkers are not responsive to this drug. So in this case, one would need to evaluate a significant or a sufficient number of studies, whereas in contrast to, for example, a cell-based biomedical research, where you might use minimum 3 or 4 biological replicates. Here we would be looking at a much larger set of samples. In the middle panel, it's a slightly different design in research, where the biomarker is first used to select certain subjects and then undergo the drug trial. And on the right side is yet another design which involves particularly more different subtypes, where the whole cohort is divided into different groups and with different stratification, then coming down to the biomarker selection and biomarker formulation. So you can see in this design, then you would need to enroll even more subjects for the whole study because you have many different subgroups. So the whole point from showing you these different clinical research study designs is to emphasize the point that we need to have very robust measurements across hundreds of clinical samples in such a study. And therefore, our proteomic techniques must be able to reproducibly measure the same set of proteins across hundreds of samples. Now, this is possibly a point that's not really carefully considered when developing novel proteomic techniques. So often, proteomic researchers designing new techniques can spend a lot of time, days or weeks, on a single sample, trying to get the very minute details and the excellent sensitivity in this technique. And however, at the end of that, it may not really be possible to apply particular measurement techniques across hundreds of samples reproducibly. And hence, in a clinical research setting, trying to measure a lot of proteins across different samples, one may need to come up with a different strategy. So that brings me to a brief overview of the key steps that we need to use in clinical proteomics research. So basically, I've broken it down to 3 aspects in the workflow. So first, there's the sample. So in terms of asking a clinical research question, we can be measuring different types of samples depending on whether we want to look for biomarkers, where the biomarker sources are, what are the available samples. And also, if we want to ask mechanistic questions, you might use tissue or biopsy samples, and possibly together with also biofluids if we're looking at proteins that are released to act on other parts of the body. And so there's a different range of samples which have different types of properties, different concentrations, and different challenges in terms of protein extraction. So that's one of the considerations in terms of the clinical research sampling. In the second part, the laboratory analysis, there are many, many different steps which could cause variability during this complex sampling process. So we can consider that protein extraction should be reproducible, protein quantitation, so measuring the concentration of your protein extract, that should be accurate. The inclusion of internal standards, the protein digestion process should be, again, reproducible, and then the mass spectrometry should reproducibly measure and quantitate proteins. Then the mass spectrometry data, I then pass on to the data analysis step. And this usually includes first the mass spec raw data conversion to protein identification and quantification. And we'll consider that in the next section. And finally, there will be the statistical analysis of the quantitative proteomics data for the clinical research questions that was asked for the entire process. So it's a big study, and one needs to really design the entire study well from the beginning to get the answers which are reproducible and may eventually be translated to a useful diagnostic test or molecular mechanism revealing drug targets into the clinic. So then that's now consider the types of quantitative proteomics techniques that are available to us. So ideally, when we think about measuring a lot of samples, hundreds of samples, we would prefer, in the ideal situation, that for each single sample, we just want to do a single run with a method that is a standard method. Then we can quantitate all of the proteins reproducibly through the entire cohort. So is it possible with current proteomics technologies? So let's consider this in the context of what mass spectrometry-based proteomics relies on in terms of its theory. So this is a very simple slide, and probably it's no news to most of you. So how does mass spectrometry-based proteomics work? So there are two steps. The first is protein identification, and in terms of the commonly used method where we are talking about bottom-up proteomics, the samples are usually protease cleaved to generate a mixture of peptides comprising all of the proteins from your sample, and the mass spectrometry data are then matched to a protein database to using different algorithms, and there are many of them available. Each of them could be slightly different, and using cutoffs which are selected to minimize false IDs, then we match the proteomics, the mass spectrometry data to the database. So from here you can see already certain restrictions that, for example, it can only identify or match proteins that are actually in the protein database. And the larger the database, there will be false discovery rate considerations, which are, a lot of this is now available, but it's when considering clinical proteomics research, we need to bear that in mind. So you should use an appropriate protein database for identifying and matching the proteins. So now, okay, since if let's say we're happy with our protein identification method, next we actually really want to get the quantitation of different proteins. And, so first before the quantitation, I would like to cover the peptide selection and identification part, because this is actually an important consideration. So how are the different peptides selected and identified? So usually in bottom-up proteomics, liquid chromatography is used to separate the different peptides. so that there's opportunity for different peptides all to be identified. And broadly, then the peptide precursors are analyzed through the first MS, so generating the MS1 spectra, and then further fragmented to get the MS2 spectra. And in a common form of shotgun proteomics, the selection of the peptide are usually based on a top number of peptides, for example, top 20 peptides. So there's a little bit of stochastic selection of peptides which eventually get fragmented and identified. So there is one set of variability in terms of protein identification and peptide identification. That one should consider that not all peptides are being fragmented during the mass spec. So now we come to the protein quantification. So in the background of what I just covered, now there are several ways that can be used for protein quantitation. And here I'm showing two most commonly used methods. So method A is to use the peak height or area of the MS1 spectra, which is measuring the peptide precursor. And this is quite accurate because there's a linearity relationship between the peak height and area for the same compound. That means the same peptide. An alternative way is to use the spectral count, so the number of spectra at the MS2 level. And based on what I explained earlier, due to the stochastic selection of the MS1 peptides that get fragmented, this possibly is slightly less accurate as using the peak height area because some of the same peptides may not be selected because they may not be, for example, the top 20 at that particular retention time for every single sample that you run in your 100 sample cohort, for example. So this is another area to consider. So to avoid this issue of the MS1, the stochastic selection, so slightly, I guess, semi-random, it's based on the intensity of the MS1. There are newer methods that have been developed to avoid this selection. So, So in this slide, just let me backtrack a little bit. So what I described earlier is the data-dependent acquisition method, which we select the most abundant ions for fragmentation. So the advantages for that is that we are able to measure large numbers of peptides in a complex mixture. But as I explained just now, there's a stochastic selection, so the same peptide may not be quantified in all samples in the same cohort. So to avoid this issue, there are two other methods that have been developed. So actually, selected or multiple reaction monitoring is a relatively old, well-established older method, so it's not really new. In this technique, the MS1 and the MS2 ions are actually already pre-selected. So we come into the assay with a list of MS1 and a matched list of MS2. So we already should know what we want to measure. And this technique gives very robust measures of the particular selected targets, and it gives good quantitation. However, it requires prior knowledge, and there is a limit to the number of targets that one can use for analysis. But it's a highly quantitative measure. So can we have a method that measures all of the MS1 and give good quantitation? So this is what the newer data-independent acquisition technique wants to achieve. So in this technique, the MS1 is broken down into blocks of independent MS1 regions, which are independent of the intensity. So the MS1 peptides are not selected based on intensity. So all possible MS data are then acquired for all your sample, but then which is good for potentially able to measure all detectable peptides. However, then the challenge is in the computational aspect, because there's in the MS2 level, a spectral library is usually needed to match and identify the peptides and then the proteins. And so the deconvolution can be rather challenging. So now we've finished considering the different quantitative methods. The data independent is relatively new. Different clinical proteomics research studies are now reporting the data and the results coming from here. The specific details of the studies needs to be considered as well when you're looking at using this method. So can there be a single LCMS method for all samples? That would be ideal if we don't really need to develop new methods for every single study. Well, that is, again, not so straightforward because the different matrix background can impact the LCMS method. So it can cause ion suppression if there are different mixtures of peptides, which you always get from every single different biological source. So, for example, a plasma sample proteome would look very different from a tissue extract proteome, and so every single type of sample needs to be separately evaluated and confirmed that there's no matrix effect and suppression. So different method of extraction, again, could cause a difference. So no, unfortunately, one should not use a single LCMS method for all samples. And then in terms of additional controls, yes, it's very good to include internal standard controls, and often this is using a stable isotope labeled peptide as a control, and later on I'll discuss additional controls that one can use. So going back to our wish list, no, unfortunately, clinical proteomics research cannot really use a single method and a single run for standard samples, so we need to really be a bit smart on how we design our studies. So after that, technical considerations for mass spectrometry. Now let's get back to thinking about the clinical cohort research design for using quantitative proteomics techniques. Now go back to this slide, which I already shared with you on the different steps that we have to think about during a clinical proteomics research study design. So let's go through them in steps. So firstly, in terms of the clinical research sampling, we need to think about the cohort characteristics. So what kind of samples we want, what are the questions we are asking. So do you want to find a body fluid biomarker that could be sampled easier, or do you really need to use a tissue sample for diagnosis because that's what the clinical practice, clinical workflows use? There are various considerations in terms of the cohort. Would you like to match the gender? Do you need to do that? It depends on your question and the diseases you are studying. What about age? Do the study subjects need to fast? And is the time of sampling important? So should one sample at the same time of day? Additionally, the processing procedures and the storage procedures should be, again, consistent across your cohort. This is particularly important if there are multiple sites of clinical sampling, so that each site, there should be a standard operating procedure across all of the sites. So that's on the clinical side. So in the laboratory and data analysis aspect, again, there are many steps which can cause different and cause variability during the sample processing. And so here, one must really minimize this technical variability. So quite often, manual protein extract and processing are used with a laboratory technician or postdoc. So even though the person may have excellent technique, just because one is extracting hundreds of samples and processing hundreds of samples, there could be variability in the sample processing. Often there's a lack of quality control measures, and we'll discuss how to deal with that. And we have already covered the stochasticity of mass spectrometry method. This we can consider by choosing appropriate mass spec methods and possibly slightly compensate through the data analysis aspect, although I wouldn't recommend really relying on doing this. So one of the stochasticity issues from the mass spec method is leading to a lot of missing data points. So in terms of data analysis, so sometimes across the subjects, certain protein measurements could be missing. So that means it was not measured. And so in this case, it may not be very clear whether or not the protein measurement is 0, or it just did not get selected in the MS1 level. So this is common in the DDA approach when MRM method is used, where a list of proteins and peptides are targeted. There's usually low missing values. And similarly, if DIA method is used, then there's again low missing values. So this is one of the points that should be considered in terms of designing the data analysis pipelines for the different types of mass spec acquisition methods. So further along in the data analysis workflow, there's QC considerations in checking that all of the different runs are good quality, the normalization methods, and again, in terms of data analysis, the different algorithms used for peptide to protein inference could cause some variability in the clinical proteomic research. In terms of that, for a single study, obviously one should use a single algorithm just to don't have a variable across different samples. So there's limited choice in that aspect. So in considering the workflow, one of the important aspects to think about is the proteome complexity in the mass spectrometry. So one of the limitations is the speed and the computational deconvolution algorithms that are needed to analyze the data. And so in a sense, one needs to separate and reduce the complexity at the peptide or protein levels to allow increased proteome coverage so we can identify more proteins. And this can be achieved in several different ways and has been achieved in proteomics research by using longer mass spectrometry runs in terms of the LC separation. You can use a multidimensional separation either at the protein or peptide level. This then leads to more different mass spectrometry runs. So again, that increase mass spec runtime. So you can see both these options increase more mass spec time, and quite often the use of mass spec time causes cost for your project. So sometimes this may not be possible depending on the budget for the research project. Apart from the cost issue, another very, very important consideration is the performance of the mass spec. So when you run a longer time, it's quite likely that the optics in the mass spec becomes more dirty due to the samples are run through, or for various reasons, mass spec performance may drop over time, and this really needs to be carefully monitored during the experiment to avoid variability that hasn't been accounted for. So going through then, if we want very deep proteome coverage of large clinical research cohorts using, for example, multidimensional mass spectrometry with long run times, really we need a very large budget for our research study, which sometimes may not be available. So how can we perform our research study in a well-designed manner, still within reasonable budget? So here are some suggestions of which we've learned through our last few years' work in this area. So in terms of the clinical research sampling, we would clearly define inclusion, exclusion criteria and to choose samples very carefully in terms of what samples we analyze. So unless the research project has large budgets, often we cannot afford to analyze all samples collected. So at different stages of the clinical research, you may want to analyze different types of samples. So through careful selection of the samples that you analyze, you can get better quality results and answer your research question. And additionally, as I previously mentioned, having standard operating procedures for sample collection and storage is very key. So we do not want samples to be already of variable quality, variable degradation, even before we get to the mass spec measurement. So this is critical, particularly for multi-site studies. Then coming to the laboratory aspects, there are several ways that one can try and minimize sample variability during processing. Very important and very useful is to use a liquid handler for sample processing. And this is not only to save the hands of your very talented and very careful research assistant or postdoc, but it actually really helps to increase the reproducibility of the sample processing. Another key measure is to spike in internal standard protein and the cis-labeled peptides, which allows you to check the quality of the runs and potentially use for normalization if required. And this helps a little bit in the data analysis aspect as well. So, but still, there are a lot of proteins in samples, in different biological samples, particularly if one is looking at plasma or serum samples, which are arguably a very common sample for biomarkers, and it's an easy sample type for example, a clinical diagnostic test. So one might want to use that as a preferred sample type for clinical proteomics research. So how do we deal about this? One of the approach we've taken is to focus our research efforts into subproteomes prior to mass spec. So this can be chosen according to a disease pathology. So, for example, particular post-translational modifications may be important in the disease, and there could be methods that are available, or you can develop particular methods to target this type of subproteome, which are post-translationally modified and relevant to your disease. And this can actually, in a single step, easily reduce the complexity of the proteome. Another example is particular subcellular organelles, subcellular locations. For example, quite popular is now the extracellular vesicles, which are released from cells, and this package proteins to send to other parts of the cell, and that could be used as an isolation method. So generally, a potential idea to reduce sample complexity that's relevant to the disease will help to reduce sample complexity going into the mass spec, and that would improve the quantitative robustness of your entire workflow. And so now I come to the question of study design. So again, if one has unlimited funds to run a lot of studies, then one could consider analyzing deep proteome for all samples, for all cohorts. However, this often is not practicable in terms of how long the mass spec will be maintaining the standard of instrument that we would like to have for the analysis, as well as in terms of the cost. So instead of doing the rectangular design, which is shown on the left of this slide, which is very often used for genomic studies, proteomic studies have often used the triangular design, which is shown on the right side of the study. And in this design, there are different phases of study, starting from the top of the pyramid, where the number of samples, so the cohorts starts with a small cohort. So in this step, one would carefully select and match the samples, the subjects, and then one may use a DIA or DDA method to measure as many proteins as possible, and I would still apply for a sub-proteome approach if possible. And after that, a set of candidates are selected through the statistical analysis, then a validation or verification cohort or qualification cohort, different terms have been applied to this, is then used as an intermediate step. And often one might design a MRM assay to very accurately quantitate the small subset of candidates. And this can then lead to an even smaller subset, although MRM assays could measure around 50 or tens of proteins. So it may not need to really go into an antibody-based stage towards the end. So a phased study design is quite feasible in terms of cost and allows a different selection and different type of proteomic methods during the steps. And so overall, what this slide is summarizing now, the different steps that one might consider at each of the stages or each of the workflow during a clinical proteomics research project. So in terms of the clinical research sampling, design the study for a focused question, and if possible, using longitudinal sampling from the same set of subjects is quite useful, because each subject will provide multiple samples and act as their own control, which takes away some of the biological variability across people. We can consider stage-relevant sample selection if the triangular design is used. So for that, what one would do is to use a well-matched sample at the start, and then go into broader selection of subjects for the later stages. And again, cannot emphasize enough the standard operating procedure for sample collection, processing and storage. In terms of the laboratory analysis aspects, I would suggest to target a subproteome if possible, include internal standard as a QC, use liquid handler for sample processing. And a very good practice, which I've learned during our research, is to actually run through the entire procedure that has been established, using, for example, a single sample run a few times to determine the robustness or variability of the entire procedure before starting the whole cohort. That way, you know what is the variability that occurs just through the sample processing. Then you can be more confident of your result at the end. So finally, in the data analysis aspect, you can definitely check your whole data set initially for overall quality, and if there are any outliers, either due to technical reason or a biological reason, those outliers should be removed from further analysis. The mass spec stability across large cohorts definitely should also be analyzed. So usually there should be mass spec QC samples, for example, often used HeLa digest, for example, or a BSA digest, just inserted during your sample sequence to ensure the mass spec performance is the same across weeks or months of runs for your entire cohort. Normalization can be used as well, and finally, one should apply suitable statistical analysis to answer the clinical questions. So now, finally, I just want to share a practical example from our research. So the example I'd like to share with you is our discovery of serum protein biomarkers for early detection of esophageal adenocarcinoma. Now, this is a cancer that develops at the lower esophagus. Most patients that suffer from this disease are diagnosed at late incurable stages, leading to a median survival rate of less than 1 year. There is a recognized pre-malignant condition called Barrett's esophagus, and the disease progression then usually occurs through Barrett's esophagus to high-grade dysplasia, which is the intermediate stage, and then progressing to esophageal adenocarcinoma. The whole procedure can be up to 10 years. There are also recognized risk factors, including reflux disease, and males, also obesity, are strong risk factors for this disease. Now, one of the clinical needs is to develop a more effective diagnostic test for surveillance or screening. Currently, the surveillance of Barrett's esophagus patients are done by endoscopy with biopsy. And this is an invasive procedure, and health economics studies have shown that the procedure is not cost effective because of the low progression rate and the long time required for disease progression. So there's a need for biomarkers which can detect early disease, meaning high-grade dysplasia stage, and for the test to be less invasive. So we aimed to discover serum protein biomarkers for this application, and we wanted to discover markers that can pick up high-grade dysplasia rather than a late-stage cancer. So to cut a long story short, we decided to focus on glycoproteins because there's literature suggesting that glycosylation is changed during cancer development in general and during esophageal adenocarcinoma development in particular. And what we decided to do was to set up a high-throughput screening method using a liquid handler. And in this method, we use individual lectins, each coupled to a magnetic bead, and array individual lectins coated with beads on a 96-well. Lectins are natural proteins, which their job is to bind different types of glycosylated proteins. So we use it as a tool to isolate different types of glycosylated proteins. This has been used quite a lot in glycobiology. The difference with our method is that we use individual lectins, and we initially selected 20 different lectins to use to screen against one serum. And in this method, then the individual serum samples are denatured, and we added a chicken of albumin to each serum sample at the start as an internal standard, so we can monitor the entire workflow by chicken of albumin peptides, which are not in any of our samples. So after that, the denatured proteins are captured at the protein level with the individual lectin beads on the 96-well. The washed proteins, the unbound proteins are washed away, and then we did an on-bead tryptic digest procedure, which then we can collect the peptides from the supernatant for mass spec analysis. And the entire workflow was set up on a Bravo liquid handler platform. So the study cohorts we used, so we chose a triangular workflow, and initially in the first stage we had 10 patients per group, and we analysed healthy, the Barrett's esophagus, and the cancer group. And we used DDA acquisition method, and we had 20 different lectins here. So we screened a large number of proteins in this method. From there, we chose 6 lectins as the most promising and designed MRM against around 50 proteins, which came out as candidates. And in the second phase, we analysed 20 patients per group. And the groups are again the same as the first phase, healthy, Barrett's esophagus, and cancer. And from the qualification phase, we chose 4 lectins, which were the most promising, and we actually just used the same MRM assay, but this time we included a labeled peptide to some of our top candidates to increase the robustness of the measures. And in the final phase, we measured a large, broad range of samples from an Australian cohort as well as from a small cohort from the US. And at this stage, we added the dysplasia samples, so low-grade and high-grade dysplasia. And the goal here is to define a biomarker signature which can detect high-grade dysplasia and cancer, labeled in red here. So these are the groups that will receive treatment, and then we want to distinguish these groups from the healthy Barrett's esophagus and low-grade dysplasia. Now, the results were of all these stages were published in these 2 studies. The final outcome is we found a 10 glycoprotein biomarker panel which can detect high-grade dysplasia and esophageal cancer as a group from the other group that don't need treatment. And the specificity in the final stage, we divided our cohort into a training cohort, which was used to develop this 10-marker panel, which has a specificity and sensitivity over 85%. Then we used that biomarker panel to test the USA cohort. And the sensitivity and specificity, as expected in this scenario, was lower than the training cohort, but it's still fairly reasonable. And this is quite promising result from these different cohorts. So finally, just remains for me to summarize what we went through today. So we consider the key aspects of variability in proteomic research using clinical cohorts and some practical solutions that could achieve robustness in the quantitative proteomics analysis of large clinical cohorts. And then I gave you an example of how we discovered serum glycoprotein biomarkers for early esophageal adenocarcinoma, and that involves the use of magnetic bead coupled lectins, and which we then developed with a multiple reaction monitoring assay, allowing validation in clinical cohorts of several hundred samples. Over the 3-month run, we achieved excellent reproducibility for the spiked-in standard, so we were quite confident of the result. So now we're looking at further developing the assay for clinical use in the future. So thank you very much for joining me today, and hopefully there are some tips that you have learned from today's webinar, and I'd be very happy to answer any questions.
Thank you, Dr. Hill, for your informative presentation. Now, as you mentioned, we will now start the live Q&A portion of our webinar. To our audience, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions already coming in. Dr. Hill, let's start with this one. Should one be concerned about batch effects in clinical proteomics research?
Thank you for that. That's a very good question, and the answer is yes. So one thing that I actually neglected to mention, and yes, thank you for the question, is that the cohorts, the samples should be randomized during the run. So that means for example, the cancer and the non-cancer samples should be mixed in order. So one should not run all cancer samples at the start and all non-cancer samples at the end. And in terms of the batch effect, that's one of the reasons of batch effect. If you are running all of them, for example, group A at the start when the mass spec is in very good condition, and then you see a drop in mass spec performance during the end of the run when group B are all run. And that's one of the reasons that one should randomize samples. And the second reason, a second way to randomize is if you have multiple microplates, for example, or different batches of processing, then one should definitely randomize the samples across different plates or batches of processing. And that means group A and group B should be in both plates or batches.
Thank you, Dr. Hill. Now, this next question is two parts. Do you have any recommendations for cohort size and selection for biomarker studies? And should samples be matched for clinical parameters across study groups?
Thank you for the question. So the cohort size is slightly variable. So it depends, again, on the number available and the study design that you go with. So I can comment more on the triangular study design, which is what we have done. So you would have seen in our initial study, in the first phase, we just did 10 samples per group. In later studies, we've moved to 20 samples per group. Although the 10 samples per group gave good results, we wanted to use more, and we had the budget to do more in later studies. So probably around 15 to 20 at the initial stage is good. Quite often, a sample size calculation may be asked for when you, for example, do grant applications. However, if you don't have any data, it's just a discovery stage. You really cannot do a sample size calculation accurately as such. So I would say between 10 and 20 per group is a good size to start with. For selection in the initial phase, the discovery stage, I would recommend to match samples very well between your groups. So group A and B definitely should be age and sex matched, and any other potential confounding factors, it's possibly good to match them so you get very homogeneous results and you get candidates going forward to the next phases. For next phases, then I would relax the sample matching criteria so you can really observe how the candidates are performing across the different types of samples that you have.
Thank you, Dr. Hill. Now we have time for a few more questions. Spiking in an internal standard protein is a good idea. Should that protein be used to normalize samples, such as one would use actin for Western blots?
That's a good question. So, indeed, I like to add in an internal standard protein to check the quality of the workflow for each of the samples, particularly when you have a cohort of a few hundred. Even using liquid handler, occasionally there may be some samples that just did not get a good quality processing, and then one could see that the internal standard peptides that were generated from the spiked-in proteins, which we're using as ovalbumin, chicken ovalbumin in the example I gave. So the use of that for normalization is good when the samples are not too variable. So if it's clear that one of the samples there was a problem with the processing, and for example you hardly see any of your internal standard peptide, then I would actually remove that sample from rest of the analysis on the basis that something went wrong during the processing and the data is low quality, rather than normalize. If there's only small variability, between samples for the internal standard protein, then yes, indeed, you can use this internal standard protein for normalization. That would be my suggestion.
Thank you, Dr. Hill. And we have time for one more question. With the invention of data-independent acquisition, will shotgun proteomics and targeted proteomics be abandoned in the future?
That's a good question. So the DIA acquisition has been used in a few studies now. It has some advantages, and I think we are still learning on all of the details on how it works. So at the moment, the other two approaches are still used quite a lot. The MRM assay probably will not be abandoned, because in the clinical laboratories, MRM assays are used to measure metabolites, and so it's a workflow that the clinical labs already use for diagnostic purposes. So in terms of translating clinical proteomics research to clinical assays, and the MRM assay is a very good bridge. So I would think that using MRM assay as that link between research and implementation would be a good idea, and that's in fact what we are aiming to do with our assay. So thank you for that question.
And thank you, Dr. Hill. I'd also like to thank our audience for joining us today and for their interesting questions. Those questions we were unable to answer today, and those that are submitted during the on-demand period, they will be addressed by Dr. Hill via the email address you provided at the time of registration. I would once again like to thank Dr. Michelle Hill for her time today and for her important research. We'd also like to thank LabRoots and our sponsor, Agilent, for underwriting today's educational webcast. Today's presentation is educational, and it does offer continuing education credits. If you are interested in receiving your credits, please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. This webcast can be viewed on demand, and you may continue to submit questions during the on-demand period. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.